Right, the second part is to give you an idea of what the evidence is for evolution. Obviously, I already explained that this is an important theory and it's the unifying theme of biology, but how do we know this? Whenever we come to these ideas, we always have to make sure that we're within science, we're doing the right kinds of things as far as science is concerned, and so evidence is obviously at the root of all this. It turns out that when you look for evidence about evolutionary processes, this is not something that we have to go very far to find. There's lots and lots of evidence for evolution. And this kind of evidence comes in several distinctive forms. And I'm grouping them in a certain way because that's what I find is easiest to understand. We have fossils, we have biogeography, we have comparative anatomy and comparative embryology, and then lastly we have molecular biology. And all of these contribute as to the whole. They're not the kind of thing that, all by themselves you can use, yes, but one of these, the fossils, can be used to test the ideas from biogeography. The molecular biology can help us test some of the ideas that we get from comparative anatomy. And so these lines of evidence together create a really nice picture of how evolution works and why it works. Now let's start with fossils. Fossils you've probably encountered before. Fossils are what's left over from an organism that lived in the past but whose existence now is only known because of these fossils. And so in most cases what you're familiar with is, let's say, dinosaur bones, and these things, these dinosaurs used to be around, and during a period of time after they died, the material in the bones, the organic material, was replaced by minerals, and so therefore they are now present as stone. Some of these fossils include entire organisms. For example, petrified trees. Now we have not too far from here Petrified Tree National Park. So there you can find entire trees, and you can see the tree rings and everything, and they're literally lying around and you can investigate. Mineralized bones, that's what I mentioned what dinosaurs are, or sometimes it's only the impressions of animals with soft tissues. Now clearly invertebrates sometimes don't have anything other than soft tissue. Well, sure, crabs have shells, and so you can find fossil shells, but with a lot of things we might not find any kind of real bony or carapace-like fossil, but all we have is impressions, for things like jellyfish, for example. And so the jellyfish was at some point in there, and it was surrounded by material, and this material now still gives us the shape of what that jellyfish looked like. When you look for fossils, the best place to find them is in sedimentary rock. Sediment is, of course, something that floats for a while and then settles. So dust, for example, as it settles, is a sediment. Or when you have the outflow of a river. The outflow of a river, as it brings all kinds of sediments with it downriver, and it settles on the ground, then that is also something that can create sedimentary rock. Now, we do have fossils where some organic residue still exists. So the idea with Jurassic Park was that there were fossils encased in amber, which is an organic substance, and you can indeed extract a little bit of genetic material from them, but we're not able to do sort of the full Jurassic Park-like experience so that you could somehow recreate these organisms. But still, fossils are pretty cool because they do tell us a lot about the past. Now here's an example for what one of these fossils might look like. This is an intermediary fossil between something that looked like perhaps a cow and something that looks like a whale. The difference is whales don't have this part anymore. Whales don't have proper hind limbs. This one still has the structures of the hind limbs. You can see here, there's basically the foot, right? There is a pelvis here. So all the different bones are there, but we don't see all these bones. In this particular instance, all we see is some of the fossils of this. So here you have some of the bones as illustrated here. So you have the toe bones, if you will. That's one and one there, and this is what you look like, what they look like as fossils. And it shows you that this is an organism that had a reduced limb compared to some of the other kinds of organisms that were there. And based on these fossils and the age of these fossils, you can come up with an idea where you start somewhere, you ended up going a certain direction, and maybe the thing that we're looking at is this is something like a cow, this is something like a whale, and what we're looking at in this picture is this. So that sometimes you can see these so-called missing links going from one type of organism to another. Now, I say something like a cow just in terms of overall body. This wasn't anything like a real cow. It's just sort of a big four-legged animal that started to live closer to the water and then went into the water. But this is how we now know the history of whales. The evolution of whales occurred, and these kinds of fossils help us with that. The fossil record itself is really interesting because we can actually trace it back through time. One of the things you'll realize is that fossils are in certain sediments based on their age. What do I mean by that? The chronology of the rock means that when you have a rocky layer down here, that contains older things than what you find up there. So the things at the bottom are always older than the things at the top. Think of a garbage dump. Where do you find yesterday's garbage? Right there is yesterday's garbage. Right here is the garbage from last week. And below that is from last month. And below that is from last year, right? So even on your garbage dump, you have a chronology of things. And that's literally the same way that a chronology is established in the rock. So you can tell that anything from down here is always going to be older than something from up there. Now, you're not necessarily able to put years to this figure at this point. There are some techniques that we'll discuss later, but right now it simply gives you a relative position of where things are from. And so the least of what this does is allow you to put things in order. It can help you understand which came first in terms of a structure and which came later. And this is, of course, important when you're trying to figure out how structures change over time. And as a consequence of allowing you to give direction to the change in which structures moved, that allows you to support other ideas of when organisms or how organisms evolved. Now, to make this a point for us here in the high desert, when you think about dust and sediment, well, clearly there's going to be dust on your vehicle at some point. So there's dust on my car. I'm going to decide to park my car out in the desert somewhere, and I'm going to leave it there for a while. Let's say I leave it there for a year. Everybody would agree that the car is going to look dusty, right? And for the sake of argument, let's just say that for that year, or during that year, I'm going to accumulate 1/100th of an inch of dust. That's not a lot of dust, but let's just say that's 1 year. Well then, in 100 years, I'm going to have 1 inch. In 100 years, this was 1 year, this was 100 years, 1,000 years, I'm going to have 10 inches. And now I can make this bigger. Now 10 inches, that's already something, right? Now at some point, once you get to 100 and 1,000 inches, well, now you realize your car is going to get flattened out. Plus, who knows what else happens? I mean, there's rain, there's storm, there's, flooding. All kinds of things could happen to that car if you just let it sit there. But the point of this is, given a lot of time, all kinds of interesting things can happen. So as long as there's time, let's not discount any possibilities. Let's just see what's in the rocks. Let's just see what can happen and go at it with an open mind. But clearly, if you are considering the Grand Canyon, for example, this is a picture of the Grand Canyon, there is probably about 1.5 billion years of history in these rocks. 1.5 billion years, and that's 3,000 feet or so. So once you have that much time, you can deposit that much sediment, and there is so much movement and change on the Earth that we actually have a lot of it in front of us. Now, biogeography is related to this, and there are some things that we'll talk about later when it comes to the movement of continents and so on. But biogeography is simply the study of the geographic distribution of species, where things are and why. This is the idea that, this is the concept that gave Darwin the idea to look for ancestors of his finches in South America. And of course, making that connection is very interesting, because that way you realize, okay, the ones in South America, there's only one kind, and the ones in the Galapagos, there's over a dozen. Now, when you try to figure out how certain species came to be in the places where they are, often the most simple explanation is the one that it's because they have a common ancestry. And you see sometimes that in areas where the animals or the organisms live quite distantly from each other, they're still very similar, and then the question is, well, did they ever at some point live together? And then something split them up. So, for example, different populations of organisms on either side of the Atlantic, did they ever fit together? And the answer is, yes, they did. But of course, it took some time to recognize that this plate movement was at the basis of this. And so when it comes to an evolutionary context, sometimes to look at where organisms live is one interesting way to find out how they're related. Natural selection ultimately is what answers the question why organisms live in the place they do live. So here's an example. Australia, as you all know, is home to a large diversity of marsupials. Marsupials are mammals, but not placental mammals like we are and most mammals are, but it's a particular group of mammals, and they have, the pouches and so on, like kangaroos and wombats and koalas. And we here have really only one kind. We have possums. That's it. We have the Virginia possum. That's what we have. So there's one species of marsupial around North America, and there are dozens in Australia. Dozens that have evolved in all kinds of different life forms, that some of which are reminiscent of some of the mammals. You may have heard of the Tasmanian wolf. That was a mammal that looked pretty much like a dog. It became extinct in the 1930s, but we do have specimens in museums of those, and they're remarkably like a dog, except that they are marsupials. So things are grouped together because of biogeography. And so the idea is that these marsupials probably came from a common evolutionary ancestor. Comparative anatomy is also a really interesting way to look at evolution, and it's one of the oldest ways by which people gather data on looking at different species. So comparative anatomy is to look at body structures. And when you do this, you realize very quickly that all these organisms you see, and however different they may be, there are still so many common threads that run through all organisms that you'll find sufficient evidence that they can indeed have come from a common ancestor. And when it comes to this, there are two concepts that are particularly important. The first one of these is homology. Homology is when we have a similarity of structures because of common ancestry. So let's say you have an eagle and you have a seagull. Those are both birds. They both have wings. And so their wings are similar because they have common ancestry. They're both birds. Now, in contrast, an analogy is the similarity of structures based on function. Now, if you look at the seagull and the eagle, it's clear that they're related because they're both birds, and that's what makes their structures homologous. But if you look at the eagle and a dragonfly, they have wings, yes, but they're very different wings, and even though they both use their wings to fly, there's a great difference between those. Now, the reason why this is sometimes confusing is when you look at structures that are similar in terms of what they look and similar in terms of what their function is, but then they're not based on common ancestry. Think about penguins and dolphins, or penguins and seals. Well, they have flippers, right? The penguins do. But those are very different kind of flippers than what a dolphin has, even though they look the same. And the reason why is because the dolphins are carnivores, they're mammals, and the penguins are birds. And so even though the flippers look very similar, they are analogous to each other. They're not homologous because they're very different types of organisms, even though the structure looks very similar. If you think of the body of the penguin, it kind of looks like it's the body of, one of the marine mammals, but it's very different because one is a bird and the other is a mammal. And yet sometimes these can be confusing. So if they are the same because, or if the similarity is because they are closely related, then we call that a homology. And if the similarity is just because of their function, then we call that an analogy. Now here are some homologous structures. These are all mammals: the human arm, the cat, the whale, and the bat, with all the forearm elements marked by color. And so what you can see here is that even though there is definitely a difference in shape and definitely a difference in how these are used, when you think about that, the human arm can grasp, none of the others can. The bat's wing is made up of the same elements than the human hand, but it can't grasp. The whale's flipper is made of the same kinds of elements, but it can't grasp and it can't fly. And so you can see that there is a significant amount of remodeling that has gone on based on the different needs of these organisms. Now think about this for a bat. A bat is trying to fly, so all these elements need to be very light. They also need to help it spread this leathery wing. And so you need to have these finger bones, that's what they would be in the human, finger bones, in a position so that they allow you to spread this out. Well, a whale doesn't need this. A whale also, as you can see here, has duplicated elements. So those individuals, when this became important, those individuals that could have a longer set of fingers for swimming, those were the ones that were favored by natural selection. So you add these little elements. Now the question is, how do you actually get to something that looks different than what we have? I mean, these two, the cat and the human, they still look pretty different, but then you have this little tiny one. How do you get something to be so short? Well, it turns out that during development, there are genes that make these bones. And if you want less of a bone, you have less time with that gene, and it makes for a smaller bone. So the idea is that these genes would be turned on longer, and these genes would be turned on longer than this one, because that's just little stubby. So there's all kinds of mechanisms, underlying mechanisms that produce this diversity, but they're still just remodels of each other. Comparative embryology is even more amazing. This idea, this concept really emerged only in the 1980s when people were able to not only look at an embryo after they've taken it out of its egg, for example, for a chick embryo, but they were able to observe it as it continued to develop, to figure out which part is added next. So rather than taking one egg and stopping its development and taking a look, now you could have the egg continue to develop, and you can continue to understand what the next step was. And so comparative embryology helps us figure out where these homologous structures come from. Now there are a couple of surprises in this, because it turns out that during our development, both you and I had at some point pharyngeal pouches, which normally would turn into gills if you were a fish, or post-anal tails. Now we don't have any post-anal tails. We don't have any tails that we can swish around the place, right? But yet in development we have them, even though they're not present in adult mammals. So that's a very interesting thing. Why would we have these? Well, we have them, and cats have them, and frogs have them, and birds have them, and fish have them. All of us have them. So it turns out that these are key indicators for why all vertebrates share common ancestry. It is having these pharyngeal pouches and post-anal tails. That's one of the set of characteristics that groups all vertebrates together and shows that we have a common ancestry. Now here, you can look at these. These are not exactly done the same way, but the point of it is, when you look at these pouches here, this is where these gill slits are, and this is where they are in us, these pouches. And if you were on a trajectory that makes you a fish, then those would develop into gills. If you are a frog and you are going to turn into a tadpole, then those would turn into your tadpole gills. But they don't in us. They simply disappear. They're resorbed and they become part of the lower jaw. The post-anal tail, same thing. There's a post-anal tail. Now, we don't need that post-anal tail for anything. The only thing we have as a leftover is a tail bone. And you don't swish with that, right? The only time when you see it or feel it is when you sit on it too hard and it hurts. So your tailbone is useless. Wait a minute. If it's useless, then why make it in the first place? It's okay. It doesn't have to be all perfect and all prim and proper. Some of these things are made because they're still in our genes, even though we don't actually use them. But the genes override their utility. The genes are just making them. And this tells you, in some ways, that everything in evolution does not move towards perfection, does not move towards a perfect preordained ending. It's just something that works blindly, and if the tail is still there, so what? We'll just make it very small and irrelevant. And this is the lesson from comparative embryology. It's pretty powerful. And then lastly, now with the way we are understanding molecular biology and how we can tie this directly into Mendelian genetics, this gave us a great opportunity to fine-tune the evolutionary relationship among species. Because with Mendelian genetics, we've been able to figure out how information is transmitted from one generation to the next, and with molecular evidence, we're now able to see what the differences are in particular genes. We can definitely determine what kinds of things are changing from one generation to the next or from one species to another. We also are able to figure out that certain genes evolve at different rates, and this sometimes allows us the establishment of something called a molecular clock. So if I look at my own genetic material right now, and I look at the genetic material of an Egyptian mummy from, let's say, 8,000 years ago, I can see what differences have accumulated over time. And that allows me to go back in time, because now I know how long it took for this level of change, 8,000 years. So if I then go back in time another 8,000 years, I can estimate what some of those changes were to lead up to the Egyptian mummy. And so the establishment of molecular clocks can be very interesting. So we can sometimes figure out when one species turned into two and what the age of certain fossils should be. These are the kind of things that we can estimate using molecular biology. Now, DNA sequences directly can also help us understand how distantly or how closely we're related, just by taking the overall picture and saying, okay, there's a certain difference here, and if it's a small difference, then organisms are more closely related than if there's a large difference. And so here is the way our family tree with the other primates work. So here on this scale you have chimpanzees, humans, gorillas, orangutans, gibbons. That's where the apes end. Apes are the primates that don't have tails. And then you have monkeys below. And you can see when it comes to all of these tailless primates, that's what we belong to, there is no more than a 5% or so difference between all of us. Less than 5%. In fact, when it comes to the humans and the chimpanzees, that difference is actually now, with the Human Genome Project, we know this, less than 2%. It's only about 1.8%. Now, that's not a lot, 1.8%. Most people, when they make measurements, they would allow measurement errors that are more like 3 or 5%. So the precision, like if you go and buy some cloth and they say, okay, it's going to be a yard long, well, they give you a little extra because they don't want to get caught in giving you too little, right? They give you a little bit extra. So that doesn't matter to them. That's a lot more what they give you than 1.8 or 2%. So this is a very close genetic match. And it's almost like it's kind of like an error in the DNA. But it's not. It happens in some particular genes that are important for development, and we'll talk a little bit about that in chapter 14. But the point of it is, it shows a remarkable degree of similarity. And if it shows such a great, remarkable similarity, that gives you a good indication that there is a very close evolutionary relationship. So these kinds of data, when you take them all together, they give you a pretty convincing picture that A, evolution is working, and B, we can pretty much figure out how it is working. And that's part 2.